Ever wonder why proteins are so important in our bodies and how they are made? These tiny powerhouses are more than just muscle builders. They're the workhorses of our bodies, playing a crucial role in everything from immune responses to nutrient transport. But where do they come from? How are they made? It's a fascinating journey starting with our very own DNA. Today we unravel the mystery of protein synthesis, a journey from DNA to protein. Imagine DNA as a blueprint for building a house. This blueprint, or DNA, is a double helix structure, composed of two strands of nucleotides twisted around each other. Each nucleotide contains a sugar, a phosphate, and a base. The order of these bases is what provides the instructions for building proteins, much like how the details on a blueprint dictate the construction of a house. Now, let's delve a little deeper. Picture a construction team ready to build a house. They can't build directly from the blueprint, right? They need a readable plan. Similarly, our cells can't build proteins directly from DNA. Instead, the DNA's instructions are transcribed, or copied, into a form that the cell can use to build proteins. This process is called transcription. So, we've established that DNA, like a blueprint, is crucial in laying out the design for proteins, but the question remains. So, DNA holds the instructions, but how do we go from DNA to proteins? This is where our first process, known as transcription, comes into play. Think of transcription as a meticulous scribe, taking down every word from an ancient manuscript, translating it into a language that can be understood by all. In our bodies, this ancient manuscript is DNA, the blueprint of life, and the scribe is an enzyme called RNA polymerase. RNA polymerase is like a diligent worker, zipping along the DNA strand, reading its codes and transcribing them into a new format. This new format is called messenger RNA, or mRNA for short. It's like a courier carrying an important message from the DNA to the rest of the cell. Now, DNA is written in a language of 4 letters, A, T, C, and G, standing for adenine, thymine, cytosine, and guanine. But mRNA uses a slightly different language. Instead of the letter T, mRNA uses U, or uracil. So, during transcription, every A in the DNA pairs with a U in the mRNA, and every T pairs with an A. C and G remain the same, pairing with each other. This meticulous process continues until the entire relevant section of the DNA strand has been transcribed into mRNA. It's like decoding a secret message, and this decoded message, the mRNA, is now ready to be read by the cell to build proteins. With our mRNA strand ready, we move on to the next step in our protein synthesis journey. Now that we have our mRNA, it's time for translation, the process of turning mRNA into protein. Think of it as deciphering a secret code. The mRNA strand, with its sequence of nucleotides, is like a coded message that needs to be read and translated. This is where our cellular interpreters, the ribosomes, come into play. The ribosomes act like tiny protein factories, reading the mRNA code 3 letters at a time. Each triplet of mRNA letters, or codon, corresponds to a specific amino acid. Picture it as a molecular game of match the pair. The ribosome matches each mRNA codon with its corresponding amino acid, brought in by a molecule called transfer RNA, or tRNA. Imagine the tRNA as a delivery truck, carrying the right amino acid for each codon on the mRNA. The ribosome links these amino acids together in a chain, following the mRNA sequence like a recipe. But remember, every good recipe starts with an appetizer and ends with a dessert. Similarly, in this molecular recipe, the appetizer is the start codon, which signals the ribosome to begin protein synthesis. The dessert is the stop codon, which tells the ribosome the protein is complete. What's fascinating is that this process happens at a breakneck speed, with the ribosome adding about 20 amino acids to the protein chain every single second. That's faster than the blink of an eye. Once the protein chain is complete, it folds into a specific shape, like a complex origami structure. This shape is critical because it determines the protein's function, whether it's fighting off a virus, transporting oxygen, or helping cells communicate. And just like that, from a simple sequence of nucleotides in our DNA, we have created a complex, 3-dimensional protein, ready to perform its role in the body. And voila, we have a protein. But why is this process so important? Protein synthesis is not just a fascinating process, it's vital for life as we know it. So, what makes it so important? Well, let's dive into that. Proteins, as we've discussed, are the workhorses of the cell. They perform a vast array of functions that, quite frankly, we'd be lost without. From catalyzing chemical reactions as enzymes, to providing structure as part of our muscles and skin, proteins are the cellular equivalent of a Swiss Army knife. But it's not just about what proteins do, it's also about when and where they do it. You see, each cell in our bodies contains the same DNA, the same blueprint. Yet our cells can be as different as night and day, from a neuron in your brain to a red blood cell in your toe. This diversity is due, in large part, to protein synthesis. By selectively reading different parts of the DNA blueprint, different proteins can be made in different cells at different times. This allows each cell to have its own unique identity and function. In addition, protein synthesis plays a crucial role in growth and repair. When you scrape your knee, for instance, it's protein synthesis that helps repair the damage. When you hit the gym and pump some iron, it's protein synthesis that helps your muscles grow and get stronger. Moreover, protein synthesis is also essential for our overall health. Many diseases, from the common cold to cancer, involve disruptions in protein synthesis. By understanding how protein synthesis works, we can develop treatments and cures for these conditions. In essence, protein synthesis is the process that brings the static code of our DNA to dynamic life. Without it, our DNA would simply be a string of letters, a book that could never be read. So next time you hear about proteins, you'll know just how they're made and why they're so crucial to our bodies.